we have quite a few people here this morning, so I guess we can get started. So today is the fifth in our series of SEBA expert seminars for 2021. And we're pleased to introduce to you, although many of you know him already, Dr. Nasrin, who has recently joined us in a new role, though, as our postdoctoral fellow at SEBA. And he joined, I think, just a few weeks ago, right? Which month you joined?
A few months, I think. Few months already, Prof.
Oh, okay, a few months already. It's just that we haven't been able to go in and
yeah.
each other. So time is sort of, it feels very odd trying to keep track of time. It's flying by without us meeting. But at least we can meet online, and I'm very happy that we can still proceed with the seminars. Sometimes maybe we get more people as well when it's online. So I'll just give a short introduction first of Dr. Nasrin. His Bachelor of Science is in molecular bioscience and biotechnology, but he has minors in environmental science and Chinese language and culture. So maybe you didn't know that about him. So maybe you can speak to him in Mandarin, I guess. And that was from the Rochester Institute of Technology in USA. And then he did his PhD in with the work that is on Mucuna bracteata as a potential platform for biofarming. And I think he's going to be giving us examples of that work in his presentation later. So he joined us as a postdoctoral fellow earlier this year. And besides the biofarming, he's interested in applications of genetic engineering to enhance crop resistance to abiotic biotic stresses, as well as plant metabolic engineering. So I will stop now and hand over to Dr. Nasrin to give his presentation.
All right. So the title of my presentations today is PlantMade Protein, Production of Anti-toxoplasma Immunoglobulin from Mucuna bracteata. So this is a portion of my PhD project. I do have some other parts of my PhD project, but that could be arranged for another seminar, I think. All right. So I would like to introduce to you to the concepts of biofarming or molecular farming. So what is biofarming? Biofarming is the productions of pharmaceutical or other heterologous recombinant proteins, such as the monoclonal antibody, the vaccine, the growth factors, or the hormones in an expression system that we can later on use for human or animals application. So there are several hosts that can be used as an expression system in biofarming. We have the animal cells, the transgenic animals, the yeast, and the bacteria as the conventional hosts. And these systems, particularly for the animal cells and the transgenic animals, are first and foremost expensive. And they are highly risky because they can harbor mammalian viruses, and they also have low scalability. And in the past 10 years, there is a growing interest in plants as the new host for molecular farming. Similar to human and animal, plants are also eukaryote. So that means plants can also produce complex recombinant proteins such as the antibody. But unlike human and animal, plants are safer because plants do not harbor mammalian viruses and they are highly scalable. So one of the most attractive factors to use plants as the expression system for biofarming is the lower cost of setting up the production facility. So iBio, which is one of the major players in biofarming, reports that they could build a plant-based production facility with 50% lower cost compared to the normal mammalian-based production facility. So because of these reasons, plant-made protein is expected to be the next major commercial development in the field of biotechnology. And the plant-made protein market is estimated to be valued at $19.4 million in 2020, and it is expected to continue to grow in the next several years. Another attractive aspect of plant-made protein is in terms of sustainability. So plant-made production facilities have reduced ecological footprint because basically you are planting plants inside there. And compared to the other hosts that generates high amount of non-recyclable waste that has to be processed in a special disposal facilities, in plant-based expression system, by right, all of the plant material that remain after product extraction are biodegradable in a normal decomposition process without the needs of an expensive and specialized disposal facilities like the mammalian-based system. And on top of that, we are only using 10% of the total protein content to extract our protein of interest, and the rest are currently being discarded. So we actually still have 90% of crude protein that are not used. So this is something that can be utilized for in the future. And plants also do not only produce our protein of interest, they are naturally producing a whole kind of secondary metabolites with commercial values that could be extracted as well in the process. And the residual biomass, such as the lipids and the lignocellulase, could also be used as biofuels. So it's a wholesome system that you could utilize everything from the beginning until to the end. Another advantage of plant-based expression system is the diversity of the plant system itself. So first of all, there are many plant species that can be used as hosts, either as a whole plant or as suspension cultures. And we can choose whether we want the protein to be continuously being expressed in several generations, which is a stable expression, which is the top left corner over there, or do we want it to be expressed only one time, or which is a transient expression system. And we can also decide whether we want to culture them in a closed indoor cultivations or we want to plant them in an open field cultivations. So there are so many possibilities of having a plant-based production system, and this flexibility allows plant biofarming to cater to the different segment of the market needs. And various recombinant proteins have been successfully expressed in many plant species, and these include model plants such as the tobacco, Arabidopsis, and we also have algae, lettuce, rice, legumes, and carrot. And in fact, actually, the first plant-made protein that was approved for human use by the FDA is the Taliglucerase alpha to treat Gaucher's disease, and it is produced in suspension cultures of carrot. It's not in this table, by the way. However, in recent years, there has been a shift of preferences for using non-food plants because there is a risk that it could contaminate our food supply chain. So having said that, these are some of the common criteria that are considered as desirable when we want to choose a plant species as the host for biofarming. And these include high biomass yield, rapid scalability, non-food crops, nitrogen fixer, and easily contained and propagated, just to name a few. So in our study, we proposed a plant species known as Mucuna bracteata as a new platform for the productions of recombinant protein. And Mucuna bracteata is a member of family Fabaceae, which means they are leguminous, they can fix nitrogen, they have rapid growth, and they have high biomass. And they are widely planted in India, Malaysia, and Indonesia as cover crop species in oil palm and rubber tree plantation. So I was usually asked these questions. In Malaysia, what do we call Mucuna bracteata? Do they have any local name? Actually, I'm not sure. I don't think we have a local name for Mucuna bracteata because Mucuna does not originate from Malaysia, actually. It originates from India. So the oil palms and the rubber plantations company actually imported the seeds into Malaysia and they germinated it over here and plant them in the plantations as cover crops. In the literature review, most of the publications about Mucuna are related to its application as cover crop plant. There are very limited study of Mucuna bracteata that applied biotechnological approach, and these are some of the very, very few publications that I could find on Mucuna application other than as a cover crops plants. So Mucuna was found to contain L-dopa, which is a direct precursor to dopamine, and dopamine is actually used in Parkinson's disease treatment. The leaf extracts of Mucuna also have anti-cancer properties, and the seed extracts have antimicrobial properties. All right, so we ask ourselves these two questions when we did this project. Can Mucuna bracteata be used to express recombinant pharmaceutical protein? And if yes, how does Mucuna bracteata perform compared to the other well-established platform for plant-based expression of recombinant pharmaceutical protein? And there are two platforms that are commonly used in plant-based expression systems. And the first one is the transient expression system that use Nicotiana bentamiana. And the second one is the stable expression system that use Nicotiana tabacum. So this is the general overall workflow for this project. And we start off with the first one, which is the vector constructions. So in the vector that we construct contain a sequence that encodes for Anti-toxoplasma IgG. So what is Anti-toxoplasma IgG? Anti-toxoplasma IgG is antibody for a toxoplasmosis disease. The toxoplasmosis disease is caused by a single cell parasite, Toxoplasma gondii. They can infect human and warm-blooded animals. And actually, it is estimated that about 25 to 30% of the world's human population is currently infected. So that's roughly 1 in 5 people are actually infected with toxoplasma, or 1 in 4 actually. Even though they are asymptomatic in healthy human, they are actually fatal for immunosuppressed patients. The current treatment is actually limited to prescription of drugs with dangerous side effects. And because of that, an immunotherapeutic intervention strategy was proposed where recombinant anti-toxoplasma can be prescribed as an alternative to these drugs. All right. So now that we have the vector that contain the sequence for the antibody, we transfer them into 3 strains of Agrobacterium tumefaciens. So Agrobacterium tumefaciens is a soil bacteria, and it has a natural capability to transfer sequences of DNA into the plant's genome. So we are using it for our own advantage in plant biotechnology research. Going on to the next part, which is the seeds germination and growth. So we germinated the seeds of Mucuna and Nicotena bentamiana in our labs, and we plant them in our greenhouse here in University Malaya. So we choose to transiently express the recombinant protein, which is the Anti-toxoplasma that I have just explained just now, through a method which is known as vacuum infiltration. So in vacuum infiltration, we use 5-week-old Mucuna bracteata, and we immerse them upside down in a liter of Agrobacterium culture that carry the vector just now. So we literally do that, take the whole plants and then put it upside down into the Agrobacterium culture. And then vacuum was applied for 30 minutes. And during this process, Agrobacterium was forced into the extracellular interior of the leaf. And after that, the plants were transferred back into the growing area where they are allowed to grow for several days before we harvested the leaves for protein extraction and Anti-toxoplasma purification. So when we conducted this project several years ago, at that time, there has been no published study yet that expressed recombinant protein in Mucuna bracteata. And in fact, last time I checked also, there has been no reports yet for any recombinant protein expression in Mucuna. So we are trying, back then we were trying to do it for the first time. So we optimized several parameters that may affect the expression level of the recombinant protein in Mucuna. And the first parameter that we look at was the leaf's position. So typically, if you look at the left picture, a 5-week-old Mucuna would have 3 trifoliates. And we would like to know what is the expression level of the Anti-toxoplasma at a different level, because in plant biofarming, there has been many reports that says that the recombinant protein productions in plants actually varies according to the leaf's location. And yes, we found it's true. We found that the bottom trifoliate actually produced 1.5-fold higher concentration of Anti-toxoplasma compared to the trifoliate at the other positions. And this is something similar that has been observed in several other plants such as lettuce, tomato, and Arabidopsis. This may be due to the fact that younger leaves at the top position have thin leaf lamina that could impair Agrobacterium infiltration efficiency. In the second parameter, we look at the harvesting period, like how many days after infiltration should we harvest the leaves. So we found that the concentration of Anti-toxoplasma that was produced by Mucuna generally decreases over time, and the highest concentration of Anti-toxoplasma was highest in the first 4 days after infiltration, which is a similar observations in onion and rose. And there are some of the factors that are commonly associated with this observation are active endogenous RNA silencing and proteolytic degradation in plant cells. And the third parameter that we look at was the Agrobacterium strain that was used for the infiltration. So we tested 3 different strains of Agrobacterium, the EHA105, the LBA444, and the GV3101. And we found that the Agrobacterium tumefaciens strain GV3101 would produce the highest concentration of Anti-toxoplasma. And it's actually quite well known that Agrobacterium strain efficiency is species and genotype dependent. So that is why it is important to test out several different strains of Agrobacterium when you are working with a new plant species. And the last parameter that we looked at was the extraction buffer that was used to extract the protein. So in the previous parameters, we used the normal phosphate buffered saline, or the PBS short form, to extract the protein. And during those times, we noticed that the crude protein extract usually yielded produced reddish brown crude extract, which is an indicator for high phenolic content that could affect protein stability. So we supplemented the PBS with several components. That is the PVPP as an phenolic absorbance, the Triton X100 as a protein stabilizer, and Triton X100 as the membrane disruptor, and glycerol as protein stabilizer. So by adding these components, we managed to get a twofold higher concentration of anti-toxoplasma compared to the normal PBS buffer. Based on this optimization, we have learned a few things about Mucuna. So firstly, we learned that the trifoliate leaves positions matters. The expression level of anti-toxoplasma in Mucuna varied according to the leaf position. And secondly, transient expression of recombinant protein in Mucuna diminishes over time. And thirdly, infiltration of Mucuna with Agrobacterium is best to be done using Agrobacterium strain GB3101. And finally, the inclusion of certain components in the extraction buffer will enhance the purifications of anti-toxoplasma. So we have also established the optimal conditions for transient expression in Mucuna, specifically using vacuum infiltration, and the process involves growing the plants until 5 weeks old, and then we vacuum infiltrate them with Agrobacterium GB3101 for 30 minutes, and then harvest the bottom trifoliate leaves after 2 days post-infiltration, and then we could extract the protein using NEB buffer and purify our protein of interest, which is, in this project, is the anti-toxoplasma protein. So now that we have established the transient expression system in Mucuna and use it to express the anti-toxoplasma, we also want to compare its potential with the model of transient expression system, which is the Nicotiana benthamiana. So we just followed the protocol set up by Leuzinger et al. 2013. So this is a very common procedure. So for tobacco, we could not do a similar vacuum infiltration method because tobacco plants are tall, and at that time we don't have a vacuum chamber that was big enough for to vacuum infiltrate tobacco. So because of that, we have to use a manual method, which is a syringe infiltration method, to infiltrate the Agrobacterium into the leaf. Even though there are different methods, but the aim is the same, which is to force the Agrobacterium into the extracellular interior of the leaf. So after the infiltration, the leaf was harvested at 6 days post-infiltration. Protein was extracted, and anti-toxoplasma was also purified in the same manner as Mucuna. So now that we have comparison of Mucuna and Nicotiana benthamiana for the transient system, we also want to look at how Mucuna perform in a stable expression system and compare it with the model of the stable system, which is Nicotiana tabacum. But unfortunately, I could not establish the protocol to generate stable transgenic lines of Mucuna. The putative transformants of Mucuna actually remain stagnant. They are not growing. They are not generating into a whole plant for a very long time. So we have to make the hard decisions to abandon that parts of the experiment. So it is actually generally well known that legume species is quite hard for a stable stable expression system. So which is what I observe personally in my own project. But we do manage to get stable transgenic lines of the model plant, the Nicotiana tabacum. It's quite easy and straightforward, and we managed to get it until the first generation, which is the T1 generation. So the T1 generation was germinated and planted in the greenhouse as well, and the leaves are harvested at 40 days old, and protein was extracted, and again anti-toxoplasma was purified from the crude protein. All right, so even though we don't have the data for Mucuna in the stable expression system, but we do have another 3 systems, and we can still make some expression analysis based on what we already have so far. So the first one, we look at the concentrations of the Anti-toxoplasma itself, our protein of interest itself. So by comparing the concentrations of Anti-toxoplasma, we can see that Mucuna bracteata, the transient expression systems of Mucuna bracteata, produce the highest concentration of Anti-toxoplasma at about 600 microgram per gram fresh weight leaf. And this, the second highest concentration of Anti-toxoplasma came from transient system as well, which is from the model plant, which is the Nicotiana bentamiana. So, but still, Mucuna produce, I think that one is twofold higher compared to the model system. And comparing the transient system of Mucuna with the stable system of Tabacum, Mucuna produce up to 5-fold higher than the average concentrations of the entire transgenic lines that I have for the Tabacum. So I have, I think in this graph, I have 26 different individual transgenic lines of Tabacum of the T1 generations. And we can see that even though they are all actually originated, they all came from the same mother plant, but each individual progenies express a different concentrations of Anti-toxoplasma, and this variation is quite huge, with the lowest concentration of 31.5 in the NT5.7 transgenic line, and the highest we could see 256.9 in the NT5.31 transgenic line. So there's a quite huge differences among the progenies themselves. But overall, still, Mucuna, the transient expression system of Mucuna outperform the model systems of both transient and stable system. And the Anti-toxoplasma was also analyzed in Western blot. So we can see that consistently there is a band that with an estimated molecular mass of about 58 kilodalton that can be detected in all of the system, in the Mucuna transient, in the bentamiana transient, and also in the stable Nicotiana tabacum. All right. However, the theoretical mass based on the amino acid sequence of the Anti-toxoplasma is actually 51 kilodalton, and that is what we should be observing, the second band that in the samples of Mucuna in the top left corner. You see there are two bands over there in the red box. So we have a second one that is about 51 kilodalton. So differences between the estimated of molecular mass between the theoretical and the end product could be contributed to glycosylation. So in glycosylation, so this is what happens in eukaryotic cells. In glycosylations, a group of carbohydrate sugar was added to the protein. So it is part of a post-translational modification, and there, it was estimated that at least 50% of human proteins are glycosylated. And since the second smaller band that we saw in Mucuna is actually around the same mass of the theoretical molecular mass at 51 kilodalton, so we suspect that these are Anti-toxoplasma that does not have the glycan components attached to it. So the glycan component could have been cleaved off either in planta during the in planta as in the cells itself, or during ex planta, which is during our extraction process, during the purification process, by one of the many plant proteases that exist in plants naturally. So proteolytic degradation in plant derived antibodies was actually frequently reported in the literature. However, there are already several strategies to overcome this problem, to tackle this problem, and this include co-expression of protease inhibitor and silencing of proteases gene. However, these strategies really depends on existing knowledge of proteases profile in the species of interest. So in this case, it is problematic for Mucuna because nobody has ever done that for Mucuna. Nobody has ever looked at the proteases profile of Mucuna. So it highlights the importance for future works to really look at the post-translational mechanism, the proteases profile and the proteolytic degradation activity in Mucuna. Even though the Western blot analysis revealed some potential proteolytic degradation activity in the Mucuna, the ELISA analysis managed to show that the purified Anti-toxoplasma from the transient Mucuna still retain its antigen binding properties at a comparable level with Anti-toxoplasma that was purified from the other two systems, the Bentamiana and the Tabacum. So as an overall comparison, Mucuna produced the highest yield of antibody through transient expression system compared to both Nicotiana, Bentamiana, and the model for the stable expression system, which is Tabacum. And we can see that generally the transient expression produce higher concentrations of recombinant protein compared to stable expression system. And in terms of time, how long does it take for us to get the antibody from these different systems? So we managed to get the antibody from Mucuna within 45 days, and that compared to the Nicotiana Bentamiana is actually 9 days longer at 54 days. So maybe 9 days doesn't sound like a significant number, but in industrial greenhouses where we can cultivate plants all year round, that roughly translate to 8 production cycles per year for Mucuna, whereas for Nicotiana Bentamiana is just about 6 production cycles per year. So the more production cycles you have, it means that you can have more outputs of the recombinant proteins, and the more outputs you have, that also means you should have more money as well. You should generate more income as well. But for a stable expression system using Nicotiana Tabacum, it took us about 7 to 8 months just to get the stable transgenic lines of the T1 generations, and it took another 1 month or 2 months before we could extract antibody from that generations. However, I do have to clarify over here that it doesn't mean that generating stable transgenic line is all bad, because in the end it actually depends on your purpose and the target market. What I mean is that we can use the transient system for rapid productions of vaccine or antibody whenever there is an outbreak. But for consistent application where we need the vaccine or antibody consistently, where there is a continuous demand for the same kind of vaccines every year, like for example, that's what's going to happen with COVID, right? There is a possibility that we may have to take COVID vaccine shot every year. So instead of repeating the entire process, infiltration process in the transient, we could generate a stable transgenic lines of plants, select elite transgenic lines that produce the highest concentrations of our protein, and harvest the seeds from that plants. And every time when we want to harvest the protein, we just germinate the seeds, let them grow, and harvest the leaves. So you don't have to repeat the entire transformation process again. So all of these factors should be taken into account in the proper cost-benefit analysis for anyone who are interested with PlantMade proteins potential. So to conclude, we have answered the 2 questions that we asked ourselves earlier. Can Mucuna be used to express recombinant pharmaceutical protein? Yes, it can. And how does Mucuna perform compared to the other well-established platforms? So in transient expression system, we have shown that Mucuna can consistently produce at least 2-fold higher concentration of recombinant protein compared to the model plant, which is Nicotiana Bentamiana. And even though we don't have we cannot get any stable transgenic lines of Mucuna, but still our transient expressions of recombinant protein in Mucuna was 5-fold higher than the stable expression of the same protein in the model system, which is Nicotiana Tabacum. All right, so in figuring out those answers, we have managed to establish the protocol to express the recombinant protein in Mucuna using specifically vacuum infiltration method. So going forward, where do we go from here? Even until now, there are many things that we don't know about these plants. There are not even any reference sequence. There are not any sequences of reference gene for Mucuna available in NCBI yet. So going back to our own project, which is recombinant protein stability in Mucuna, we should really readdress the issue. Is the structural heterogeneity that we observe was really because of proteolytic degradations of the glycosylation chain, or is it because of something else? If it is because of proteolytic degradation, how could we overcome the problem? Should we make a proteases profile of Mucuna? Could we create a Mucuna cultivar with lower protease activity perhaps? Um, can we produce and extract recombinant protein from at least 1 kilogram of Mucuna leaves? How do we process the leaves, and how do we ensure that recombinant proteins are stable during the extraction process? Because for our projects, we are only working with several grams of leaves, so that's quite small. But in industrial scale, they could process up to hundreds kilograms of leaves at the same time. So how do Mucuna perform in that system? We should also evaluate the efficacy and potency of recombinant protein from Mucuna in vivo in the real animal model. And for those of you who are working in plant biotechnology field, we all realize that at this point, having a reliable procedure to generate stable transgenic line is very essential in any plant study. So it is important to establish a reliable protocol to generate stable transgenic line of Mucuna, because this protocol can then be used to facilitate future study of Mucuna in genomic, transcriptomic, proteomic, or even metabolomic. And last but not least, a proper techno-economic analysis to really evaluate Mucuna potential in terms of its true industrial setting in biofarming. All right, these are some of the references that I use, that I cited for this presentation. And I would like to leave my presentation by sharing some information with some of you. I hope some of you are more interested with plant biofarming now. So there is an upcoming conference specifically for plant-made proteins. So the host is universities in Japan. Some of the speakers are actually CEOs of some of the well-known plant-made proteins companies in the world. So you can scan that QR code over there and register. And no, I do not get any commission to promote the conference. I'm just sharing it for your information. All right, that's all. Thank you very much and stay safe.
Thank you very much, Nazrin. Very nice work and a nice presentation. And I see a pop-up to just remind everybody to go to the link in our chat box to fill in the form. So we now open the floor for questions. Dr. Najia, please go ahead.
Hi, Dr. Nazrin. Thanks for the clear presentation. I realize you mentioned about the different expression level when you inoculate. I think the when you when at different position of leaf. So can you explain a little bit more on that based on your experience doing that? Thanks.
Yep. All right. Thank you, Dr. Najah, for the questions. When we started looking at plant biofarming and looking at what are the parameters that we should actually look at. So when I read the publications, so leaf's position is usually one of the factors that researchers look into when they are looking at recombinant protein productions. So in my study, we do observe that the highest concentrations of protein is at the bottom leaves. So in Mucuna, the bottom leaves is actually the oldest leaves, whereas the top leaves are the younger leaves. They are forming from bottom to the top. So we observe that the oldest leaves have the highest concentration and the concentration decreasing as you get to the younger leaves. But that is true for Mucuna and some of the species. Whereas in other plant species, the other way happening around. So there are, of course, when you read like 10 papers, all of the 10 papers will suggest 10 different explanations why such observations occur. But in my mind, I have been thinking about this for such a long time that for Mucuna, a logical explanation, for me, I think it's logical that in older leaves, because it is a mature leaves, it contains the older leaves contain mature cells, so they have a mature protein machinery systems inside of the cells itself. Whereas the younger leaves, they contain a very active protein machinery system where they, at that point, because they are still young leaves, they are still need to grow. So most of the protein machinery production system is geared towards growing the leaves upright. So there you are inserting a genes for antibody that actually is not needed for the plants. So there is some competitions going on over there. Should the cells produce antibody, which is actually what they don't want, or should they actually focuses on producing the important proteins, the mitochondria or the actin or the whatnots for them to grow. So I believe that in younger leaves there is quite competitions of materials inside of the cells itself. So this may affect the expression levels on top of the other uh, reasonings that was proposed by other researchers, such as the RNA. That's not RNA silencing. Yeah, on top of the anatomical structure of the leaf itself. Yeah, so that's my opinion of why we could observe some differences between the old leaf and the younger leaves. I hope that makes sense, Dr. Najah.
It is make a lot of sense. Thanks
All
so
right,
much. Right.
thank you.
Thank you. Prof. Suba, you have a question?
Hello, Prof.
Hi, Nazrin. I've been following your work since I think you started your PhD and
Yes, Prof.
in
You were there since the beginning.
Yeah. So it is wonderful to see that you have got good results, and of course it can be used for industrial application. The reason I'm interested is because we have a problem in our lab. We are trying to express transmembrane protein. And when we do it in the bacteria, it's not expressing itself because the membrane is actually a transmembrane protein. We have been troubleshooting for almost 1.5 years. So I was just thinking if using this transmembrane protein, it's actually a lectin which we isolated from the prawns. Can it be used to be expressed in your system? I mean, the whole team system lah, Prof. Yasmin, Prof. Jenny, and everybody's system. So I was just thinking, do you think we can try it out? Because I'm just thinking because we can actually feed the prawns the leaves. Freshwater prawns are actually they're actually vegetarian. They have they can actually eat the leaves. And I was just thinking in that line. What do you think, Nazrin?
Yeah, Prof. The transmembrane protein is plant's protein or
No, it's a prawn. Prawn
it's a
protein.
prawn. Oh, okay, udang. Okay, all right. Okay, so and you have been trying to express it in a microbial system, right?
Yes.
Yeah. So my take would be it is highly advisable to express it in eukaryotic systems at least. So you can actually do it very quickly using tobacco syringe infiltration. So you just construct some vector, plasmid dicambia, easily available in all of the plant research labs in I think. And then just quickly transform into Agrobacterium and then prepare several mils of suspension, and you can syringe infiltrate in tobacco. So
Nazrin, we collaborate, Nazrin. I mean that's what we are here for. So I hope we collaborate. I have a current intern with me, Roshini, and I've already told her to read up. And if that is, why don't we give it a try? Then later, as a team, we can actually apply a grant to further this. What do you think, Jenny? I want to give it a try, see whether it works or not.
Yeah. It's always only a matter of money and time. But if there's a will, there's a way. I know that someone else put a question before Prof. Yasmin, but I'm going to invite Prof. Yasmin first. Rossini, I'll come to you in a minute because I think she has a comment on the current question. Go ahead, Prof. Yasmin.
Hi everyone. Thanks, Nazrin.
Hello, Prof.
Presentation. I'm very excited to hear Prof. Shuba's question because lectins are naturally expressed in plants, and lectins from plants has the extraction protocol is very well established. But what's more exciting is Mucuna. It could be an edible vaccine for prawns, right? Because this is a leguminous leaf which people use for animal feed. So this could be kind of the ideal marriage. Go for it, Nazrin. I think this one you can raise funds because it has such a practical application. Yeah. So there may be investors in the audience actually, because I know that some of external visitors are here today. So this is one of great opportunity actually. Hopefully
Yeah, that would be great, and I do hope they will contact. They can get in touch with Sabor or directly to Dr. Nazrin or Prof. Suba to discuss this. This would be like Prof. Suba was suggesting, it could be done probably quite quickly and not too expensively, and is certainly worthwhile looking into. Okay, I'm gonna. Rossini, do you want me to read this out, or are you going to come on mic and ask yourself?
Prof, can you hear me?
Yeah, go ahead.
All right. So, good morning, Prof, and also good morning, Dr. Nazrin.
Good morning.
So, first of all, I really like your presentation. It was very good. It was
Thank
very
you.
clear. I really feel that it was like straight on the point. So it makes me more understandable on your topic. And I have two questions from your presentation. One is that I found that you use two different modes for your transient expression, which was you were using the vacuum and also the syringe infiltration. So why do you actually choose different mode of infiltration instead of using one for both organisms? And how does it actually help for your research, and also how if in case you would like to have a further studies regarding using this Mucuna bracteata, would you use the same mode of infiltration or would you try something else new?
All right. Okay. Thank you very much, Rosini. And I was just reading your questions in the chat. Those are the first questions, and the second one,
Yes.
Mucuna was mentioned to have several properties, anti-cancer. Is there any study then? Okay. All right. So I will answer the first questions first. Thank you very much for that question. I actually suspect that someone might ask, why do I use a different system between syringe infiltration and Mucuna? So I would like to share a page from my thesis actually, because I have the picture inside. So actually before I did, because I did not include this in my presentation just now. So we did actually do what do you call that? Like a prior study, like testing out. Can we do a syringe infiltration in Mucuna? Because obviously it is actually much easier for a lab scale operation compared to doing a vacuum assisted infiltration method. Because for vacuum, you have to prepare like 1 liter of Agrobacteriums, whereas for syringe, you only have like 10 ml of agrob, and that's more than enough. So I actually tried to syringe infiltrate Mucuna. So if you can see the pictures on the A is one leaflet of the Mucuna. So in the B, so you can see that tiny dots over there, the dark green patches over there. So it is actually it is actually what it looks like when I try to syringe infiltrate the leaflet of Mucuna bracteata. So in syringe infiltrations, the problem is the Mucuna bracteata leaves is quite fragile. They are brittle, more specifically brittle, and they can be easily damaged if I apply too much pressure. So even when I managed at least to infiltrate a very tiny portions in the leaves, the infiltrated area did not expand beyond the syringe nozzle area. So compared to Nicotiana bentamiana, it is very easily. Once you manage to infiltrate, it will disperse into the entire leaves. So it is very straightforward procedure. So that is why we have to apply a second procedure, which is using a vacuum-assisted infiltration, and you can see that it does a much better job to infiltrate the Agrobacterium into the leaves, as demonstrated by the dark green patches of the leaves. Yeah, all right. So basically, that explains why I do a syringe why I do vacuum infiltration of mucuna and not syringe infiltration. And for the tobacco, why I do the other way around, because we don't have a big enough vacuum chamber that could fit tobacco plants. So if we do have such big, so that would be nice to compare this using the same method. But unfortunately, we cannot do that. So that is why there is a differences in the method. So the follow-up is, if I were to repeat this experiment in the future, which method would I use? So for mucuna, actually, there is no easy ways of doing it since we cannot do syringe infiltration. So whether I like it or not, vacuum infiltration is the way to go at the moment. But of course, personally, I like to try to generate a stable transgenic lines of mucuna, because in generating stable transgenic lines, the difficult part is only to get the transgenic lines. But once you have the transgenic line, you can harvest the leaves indefinitely as long as the transgenic lines lives. So you have an endless supply of leaves. But if you are talking about in commercial settings, like if I want to if I want to extract like 1 kilogram of leaves, how should we do it? So you cannot do syringe infiltration. You cannot go to one by one of the leaves and infiltrate. So vacuum infiltration is the method to go in the current commercial settings. So you just dump the entire plants in one big suspension Agrobacterium, and it is more efficient that way. All right, so the second one is about cancer properties. Yeah, so the first the paper that I cited earlier was actually quite old, was actually several in the early 2000s. I'm not sure if there is any follow-up study by that group. But I think even if there is, it's actually quite limited. And I'm because I'm not from cancer background, so I could not answer what type of potential cancers can be used with there. Yeah, sorry about that.
Okay.
Something to
We
explore.
have a question here from industry. So I'd like to invite Mohammed Razwan. Do you want me to read that, or do you want to come online? Is Mohammed
Hi,
Razwan?
Nazreen, everyone. Hi, everyone.
Great.
Hello.
Hi. Thank you for this event. I think it is very good. I'm working in industry whereby we process the biomass from the agricultural waste. So I see there's a lot of biomass produced from this particular plant, and it is quite interesting that they have the nitrogen fixing on it. When looking at your slide, I think one of your slide, you were saying that 10% would be potentially to be produced as the agroprotein product of yours, and I see that 90% of that is the byproduct of the entire process. So don't you think 90% is high? Because right now when we do in our production, we are considering that even 30 to 40% is quite high for us. And when we have high percentage of biomass or high percentage of waste, then we will need to incur waste management costs on that. What is your perspective on that?
Yeah, definitely. I agree, Encik Rizwan. So that is actually one of the points of discussion in the paper that I cited, that at any point it is very challenging for expressions of recombinant proteins in plants to get as high as 10%, 20, 50%. So that's very the current challenges. So they are trying to increase the recombinant protein concentration through various methods. So most of them actually depends on the molecular constructs, how you design the gene sequences, how you design the vector constructs. I think they have some success in it, but still, it is the concentrations of the recombinant protein are quite limited, and that is one of the challenges that still persists until today. But definitely, if we could eliminate, we could optimize, produce as high as possible. That will be great, as you see it from the industry perspective, and definitely the byproducts, the biomass, all of the secondary metabolites, all of those can actually be utilized for other commercial aspects. I believe, instead of being thrown away like that.
Yeah. Yeah. On my views that perhaps you already have the process to optimize for your product. I would say that it seems that if this project is to go for to raise a funding for future, future exploration in the industrial scale or pilot scale, I would say that your techno analysis would be very helpful for us. And the other part is we would be interested to see at your life cycle assessment, on the entire biomanufacturing plant. And this will trigger the industrial interest to further explore. Because we understand that for pharma, sometimes, yes, I think 10% is actually quite good because the price is actually very awesome for us to compensate what we have left in the 90% of the byproduct. So I think the focus is now is actually for you maybe to focus on the 10%, how you optimize the 10%, and maybe a purified protein of that, and the life cycle assessment, and looking forward for your technologies will be very, very good. Thank you.
Yeah, thank you, sir.
Yeah,
A lot of optimizations to be done. It's a new point,
those are very important issues that always a bit of a gap between the sort of research we might do for a thesis and then bringing it not just to be a thesis sitting on a shelf, but to actually get it to be something that is out for our biotech community. So very good points there. Thank you, Razwan. So we're going to close it up now. We will have more seminars in the future, so please look out for those on our Facebook and Twitter. And stay safe, everybody, and we hope to see you all again soon.